This is the first lecture for Chapter 15, Microbial Mechanisms of Pathogenicity. In the previous chapter, we focused on disease in a population. For this chapter, we are going to focus on how a microbe causes disease in one individual. For a microbe to cause disease in one individual, there are several things that have to occur. The first is that the microbe has to go through its preferred portal of entry. Once that occurs, then you need sufficient numbers of the microbe entering the person's body. Those microbes have to be able to adhere to the tissues in the person's body. Then those microbes have to evade the person's defenses. Once all of that is done, then the microbe can start causing damage to the host cells, which results in the actual disease. And the last stage is that the microbe has to leave by the portal of exit. Often, before this process can begin, the person himself or herself has a predisposing factor, which makes them more likely to acquire a disease from this infection. And as a review, some of the predisposing factors that I went over already. Gender. Gender can have an effect on how the body is structured or the hormones and how they affect the immune system. Genetics can predispose an individual to a particular disease. Climate, or the time of year, like in the winter when the air is drier, people are predisposed to respiratory diseases. Age. Your immune system is not fully developed until you're about 7 years old. So before the age of 7, you are immunocompromised. And then, of course, as you age, your immune system does not function as well as it should. Nutrition. Having poor nutrition can affect your immune system and how it functions. And, of course, things like smoking and drinking can have an effect on your immune system's function. So once you have a person who is probably predisposed to a particular disease, then the microbe has to enter that person's body. And again, I've already talked about different portals of entry. The most common portal of entry are the mucous membranes, and the mucous membranes are found on various parts of the body. Most common is the respiratory system because you are breathing many times per minute. Then next is the gastrointestinal system because you eat or drink several times a day. Then you have the urogenital system. And lastly, the conjunctiva. Conjunctiva, those are the mucous membranes around the eye. The second most common portal of entry is the skin, just because it covers the entire outside of our body and comes in contact with the environment. And then the least common portal of entry is the parenteral route, where microbes are injected beneath the skin, either by an insect or it could be a hypodermic needle. Microbes don't just have to use a portal of entry. For the most part, they need to enter via their preferred portal of entry. So for most microbes, in order to cause a disease, they need to enter the body a specific way. One example is picornavirus. Picornavirus is the group of viruses where one strain causes the common cold. In order to cause the disease, the common cold, that virus needs to enter the respiratory system. If you take the virus and just rub it on the skin, then you will not develop the common cold. Vibrio cholerae is the bacteria that causes cholera. And in order to acquire the disease, cholera, you need to ingest the bacteria. So it needs to enter the digestive system. Again, if you rub that on the skin, you are not going to get cholera. If it just remained in the upper respiratory system, you would not develop the disease cholera. Yersinia pestis is actually an exception to the rule. Yersinia pestis has 3 portals of entry: the parenteral route from a flea bite, respiratory system inhaling the bacteria, or the gastrointestinal system ingesting the bacteria. All 3 of these preferred portals of entry will develop into the same disease of plague. E. coli. E. coli may seem like it has more than one preferred portal of entry, but really for each disease E. coli produces, it has one preferred portal of entry. If E. coli enters the urogenital system, it will cause a UTI. If instead the E. coli enters via the gastrointestinal system, then you will have food poisoning. And then also if E. coli is injected into the circulatory system, into the blood, then you would have sepsis. So E. coli can enter different portals of entry, but it will cause different diseases depending on the portal of entry. If you ingest E. coli, you will not get a UTI. This is a table from your book which correlates different portals of entry and shows you which pathogen uses those portals and the disease the pathogen causes. And it starts with the most common portal of entry, the mucous membranes in the respiratory tract. And if you notice, most of these are respiratory diseases, of course. Then the gastrointestinal tract is the second most common. And these diseases are all caused by ingesting the microbe. And of course, the urogenital tract, those would be the STDs. And then the skin and parenteral route are less common, the mucous membranes. And so all of these are entered that way, either on the skin or by injection, parenteral route. So in this process of having a microbe cause disease in an individual, you start with an individual who has a predisposing factor. The microbe enters through its preferred portal of entry in order to cause a particular disease. And the next thing that's required is that you need sufficient numbers of those microbes entering the individual to cause disease. Logically, the higher the number of microbes entering an individual's body, the more likely disease will occur. But they have actually experimentally determined some numbers for some of the microbes. The first type of number is called the ID50. ID stands for infectious dose, and 50 relates to 50%. So this is an experimentally determined number where the number of microbes causes 50% of the test organisms to become sick. So it's the number of microbes that causes 50% of the test organisms to become ill with the disease. The LD50, this stands for lethal dose 50%. And this can be used for bacteria, it can be used for viruses, it can be used for toxins or radiation. It's the amount of the pathogen or the substance that causes 50% of the organisms to die, so lethal dose. So LD50 can be used for bacteria, viruses, toxins, or radiation. This is another table from your book, and this gives the estimated ID50s for different types of diseases. And as you can see, measles has the lowest ID50. So half the people who inhale one virus particle of measles will acquire measles. Cholera is at the other end. Cholera, in order to have 50% of the people develop the disease cholera, they need to ingest 100 million bacterial cells. All right, so I've talked about microbes entering their preferred portal of entry. Then they have to enter in sufficient numbers. They have to meet or exceed the ID50. And once those microbes are in the body, they then have to be able to adhere to the person's tissues. Because if the person inhales the measles virus but immediately exhales it out, then measles, the disease, will not occur in that individual. So attachment or adherence. Once the microbe has gained access to the body, it has to attach or adhere to your tissues. And for this part of the chapter, we're going to go back to the structure of the different microbes that I talked about at the beginning of the semester. So for bacteria, if you remember, which appendage was used for attachment? Hopefully you came up with the answer of fimbriae. Fimbriae are these long hair-like structures, not as long as flagella, but long hair-like structures that extend from the bacteria, and they're used for attachment. I also talked about a layer of the bacterial cell that helps attach bacteria to surfaces, and hopefully you remember that it was the capsule. So the capsule is this white area around the bacillus cell right there. So that is the capsule, type of glycocalyx. Glycocalyx stands for sugar coat, and remember, when sugar gets wet, it's sticky. So having fimbriae, having a capsule, would allow the bacterium to adhere more strongly to the tissues if it gains access to the body. Okay, now viruses. So when we talked about the viral life cycle, the very first stage was attachment, where the virus recognizes and adheres, attaches to the appropriate host cell. So for the naked viruses, what was the structure they used to recognize and adhere to the correct host cell? It was these protein fibers that extend out from the corners of the naked virus, the non-enveloped virus. And then for the enveloped virus, which structures were used for attachment, recognition, and attachment? The spikes. So those protein spikes that are extending out from the envelope, which remember is a phospholipid bilayer. So the microbes have found an individual with predisposing factors. They've entered through their preferred portal of entry, they've met their ID 50 number, and they have structures that allow them to adhere to the person's tissue. So now that the microbes are inside, associated with the tissues of the individual, now they have to evade that person's defenses. And this is a preview into the immune system because, of course, your immune system, one of its main functions is to protect you from infectious disease. So when a microbe enters a person's body, there are three parts of the immune system that it needs to worry about initially. The first thing it needs to worry about is phagocytosis. There are certain white blood cells in your body, and we will talk about them later, and their job is to phagocytose, to ingest any microbes that enter your body. The second defense that they have to worry about is fever, and the third defense are antibodies. And antibodies are serum proteins that are found in your blood. Some bacteria have structures that help protect them from some of your initial defenses. And one of the main protective structures in bacteria is the capsule again. So again, here is that white capsule surrounding the bacterium. And the capsule again is a type of glycocalyx. And the capsule helps the bacterium avoid being phagocytosed by white blood cells. Other structures that can help bacteria avoid your initial defenses can be found in the cell walls. In certain bacteria, they may have M proteins. M proteins are a type of protein that is associated with the cell wall. So it is associated with that peptidoglycan. And M proteins are protective in a couple of different ways. They can help the bacterium avoid being phagocytosed. They are also heat resistant. So if a fever occurs, that helps protect them from the increased temperature, and they are also acid resistant. So where in the human body would a bacterium encounter acid? In the stomach. So for those bacteria that contaminate the food, they will be ingested, they will go to the stomach, and encounter the acid, and if those bacteria have M proteins, it helps protect them from the acid in the stomach. Another component that you can find in certain cell walls are waxes or mycolic acid. So mycolic acid, this is only found in the Mycobacterium. Mycobacteria leprae, Mycobacteria tuberculosis. So remember the mycobacteria, they are separated out from the gram-positives and gram-negatives because they have mainly waxes and mycolic acid in their cell wall. And these bacteria, the bacteria that cause leprosy and tuberculosis, they are very resistant to phagocytosis. So these waxes and mycolic acids make those bacteria highly resistant to phagocytosis. Besides certain structures, some bacteria have exoenzymes that they make and secrete to protect them from some of your defenses. So these exoenzymes are produced by the bacterial cells and secreted out. So these bacterial cells need to be alive so that they can express these enzymes and secrete them out to protect themselves. In general, the exoenzymes can be divided into two groups. One type dissolves materials. The second type has an effect on blood clotting. So again, exoenzymes, they're called exoenzymes because they're made inside the cell and they are secreted out. Two exoenzymes that dissolve materials are the first, hyaluronidase, again an enzyme ACE, hyaluronidase. It is an enzyme that breaks down hyaluronic acid, which is found in connective tissue. The second one is collagenase. So again, ACE, it's one of the exoenzymes, and it breaks down collagen, which is also found in connective tissue. So hyaluronidase, collagenase, both of these allow the bacteria to break down the connective tissue to facilitate spread. But breaking down this connective tissue will kill the tissue and it leads to necrosis or gangrene. And one of the most common bacteria that have both of these types of enzymes is Clostridium perfringens. And Clostridium perfringens causes gangrene. So when the bacteria enter the body through a cut or a puncture, they start secreting these enzymes so they can spread through the tissue and get some nutrients, and that causes death to the tissue. Other exoenzymes can have an effect on blood clotting. The first is coagulase. Again, ACE is an enzyme and it causes the blood to coagulate. So it forms clots. And this is a benefit to the bacteria because it can form a clot around the area it's infected to prevent white blood cells from getting to it and trying to kill the bacteria. The other type of enzyme that has an effect on blood clotting is kinase. ACE again is the enzyme, and these are enzymes that break down clots. So sometimes it's to the bacteria's advantage to break down a clot so it can gain access to the circulatory system. And two benefits for getting into the circulatory system. One is now that the bacteria can travel throughout the body. It can spread throughout the body. The second benefit is now the bacteria can get the nutrients that are in the blood. The last type of exoenzyme I want to talk about is IgA protease. So IgA protease, again, ACE, it's an enzyme. IgA is an antibody. So IgA is an antibody, and antibodies are proteins. So this is an enzyme that breaks down IgA. And this is a diagram of IgA. Now we haven't talked about antibodies, and we won't for a couple of weeks, but just to let you know, this antibody's main function is to prevent attachment. So this antibody is found in all sorts of secretion. It's found in the mucous membranes, and its whole purpose is to prevent the microbes from adhering to your tissues. So if the bacteria is able to secrete this exoenzyme, it can break down the IgA, and then the bacterium is more likely to be able to adhere to your tissues. So far for this lecture, I've gone over the disease process of how microbes will cause disease in an individual. So if you start with a person with predisposing factors, the microbes have to enter through their preferred portal of entry, again in sufficient numbers to meet the ID50. Those microbes have to be able to adhere to the tissues and then evade the person's initial defenses. And that's what I've covered so far today. Next time, I will talk about the specific damage that those microbes can do to the host cells and then the portals of exit.